I'd like to welcome everyone to the launch, the formal launch of the tranSMART platform version 1.2. Um, we're joining here by webinar coming from the University of Michigan campus in Ann Arbor and distributed throughout the globe for our various demonstrations. This agenda is really to formally announce the, the launch of the 1.2 platform, which is something that we've been working on as a community for the past nearly 12 months. Just for everyone's information, the events are being recorded for future offline viewing, and those events will be made available on the Foundation YouTube channel at some point in the near future. The agenda for the program today, I want to give you a few opening remarks and set the stage for what you're going to see. But we're going to go through basically the highlights that we have of version 1.2 and how 1.2 really sets a new mark for the tranSMART platform. The release highlights will be given by Brian Athey today out of Ann Arbor, who will be going, he's the Chief Scientific Officer and co-founder of the foundation, and he'll be going through some of the key features and introducing the demonstrators for each of the key features that we're demonstrating. At that point, we'll run through some demonstrations from the key developers of these key capabilities. So we have 7 individual demonstrations, which is a subset of the functionality that is new to version 1.2. But you'll get a good flavor for all of the key features that have been brought to bear, and you'll be getting that information directly from the groups responsible for developing that capability. As we go through the demonstrations, we'll get to the point where we've completed that, and Rudy Potenzone, who's the VP of Marketing for the Foundation, will give us an overview of the documentation and the video demo programs that we've put in place, and then take you through rebranding. What you'll notice on our slides today is a new logo and a new look and feel for the Foundation, and Rudy will be bringing you through that and telling you about what we're doing in the near future. We'll have a little bit of time at the end for questions and answers, and then I'll give you some quick closing remarks and tell you a little bit about what to look forward to in the future. With that, I'd like to give you the quick introduction to this presentation and to this demonstration. The tranSMART Foundation is about 1.5 years old now. It is the second iteration of the tranSMART Foundation. It was co-founded by Brian Athey and Michael Braxton-Taylor, working with E.K. Go, Kevin Smith, myself, and a number of others. And the real vision that united us in forming the tranSMART Foundation was to realize the promise of translational research, and that is to really make effective the translation of research and discovery research in particular, and to realize that impact on the clinical practice of medicine. That translated for us into a mission statement specifically around tranSMART, and what we're really focused on as a foundation is enabling the sharing, integration, and standardization of heterogeneous data that comes from collaborative translational research. The nature of translational research is that it is a group and collaborative activity, many times distributed globally in its scope. And the goal of the tranSMART Foundation is to work with the tranSMART platform, an open source and open data platform, to build a community that can more effectively collaborate in this area of translational research using the tranSMART platform as an enabling technology. To give you a sense of who the Foundation is and what we do, the management of the Foundation are members of industry, both pharma and biotech, as well as service providers and software, coming from academia as well and nonprofit sectors. And we are a team of experienced managers. There's a lot of gray hair around the table who come together to spend part of our time devoted to the goals and the mission of the Foundation. The board of directors of the Foundation is drawn from our members. We are a member-driven nonprofit company. They represent industry, academia, government, and nonprofit sectors. We have a 15-member board that governs the overall activities of the Foundation. And then as a member-driven corporation, a foundation, our members really are drawn from across the translational research community, from pharmaceutical companies, biotech companies, academic medical centers, service providers, nonprofit, and research foundations. And these are groups that come together to support the Foundation and the developing community around the Foundation. And of course, most important is the community. The translational research community is really composed of those people involved in early discovery research through clinical proof of concept. And it really spans the gamut from pharma, biotech, and academia, as well as involving key government and government-sponsored research programs. I want to introduce the management team very quickly, just so you know who we are and who will be speaking with you today. I'm the CEO, Keith Elliston. I'm a veteran of pharma, coming from Merck and Bayer, biotech, where I spent 15 years doing biotech startups. And then the last 3 years working largely in the nonprofit sector, initially at CHDI, the Huntington's Disease Foundation, and more recently with Orion Bionetworks and others. Kevin Smith is our Chief Operating Officer, Secretary, and Community Manager. He is an academic software veteran out of the University of Michigan and NCIBI, amongst other programs. Our Treasurer and CFO is Ashley George, who's a pharma veteran. Coming from GSK and also the nonprofit sector of Pistoia Alliance. Brian Athey is our Chief Scientific Officer. He's a true academic thought leader in the field of bioinformatics and computational biology. He's a department chair at the University of Michigan and was the key leaders behind the NCIBI program. EK Go is our CTO. He is both an academic and industry veteran, having been very early and thought leader in Imperial College in the areas of computer science and data science, as well as forming software companies, including InfoSense, which has been incorporated into IDBS, where he's the Chief Innovation Officer. Rudy Potenzone is our VP of Marketing, another true industry veteran coming from the software side, working with Ingenuity, where he headed up marketing with Microsoft, Cambridge Soft, and PerkinElmer. And then Michael Braxton-Thaler, again, one of our co-founders, who's in charge of our marketplace development and working with this, is experienced both in pharma and in the nonprofit sector, working with Roche and also being the CEO of the Pistoia Alliance. And so what we brought together is a very experienced team that spends a key part of its time focused on the goals and the mission of the foundation and works with the community to realize this in the efforts that we put forward in terms of the platform that you see today. The goals that we've enumerated and that this version 1.2 is a key deliverable of has been to establish and sustain tranSMART as preferred data sharing and analytical platform for translational research. And I think that we've really seen that blossom over the past year. We think that the version 1.2 will really meet a lot of the needs of the community and will establish that even further. Our goal is to link academic, nonprofit, and corporate research communities together so they can share data, share information, and do collaborative research facilitated by the tranSMART platform. We also want to align and grow a vibrant developer network around the scientific goals of the community to ensure that we can continue to innovate this technology that's really needed in this space. And then finally, we want to focus on reducing the barriers to entry through use of advanced technologies in an active marketplace. And what you'll find with version 1.2 is that we've done this in terms of the ability to install the platform, to utilize the platform, to make use of it. We believe we've really reduced the barriers to entry. And that's a real critical aspect in growing the adoption of the platform through our community. But the real focus of the foundation is growing the community. The community is what makes the platform enables the community, but it's really being, growing, and participating in the community that is the goal of the foundation. And it's this community that has come together to both develop and to test and to deploy the platform that you'll be seeing today. So one of the questions that is obvious to ask here is, well, why tranSMART? Why is tranSMART of interest today? And it's really of interest because of the growing problems that we see in translating research, basic research, into therapeutics in the clinic. One of the things that we know is the cost of developing a drug has gone from about $100 million back in the early 70s to, in some estimates today, over $13 billion per new drug, depending on the company developing it. What's really interesting as well is that we've seen an increase in the rate of phase 3 failures. When you've invested the most in a drug and you get to the point where you're really doing validation of that efficacy, still 50% of phase 3 trials fail. This is really the key that we are trying to enable. And why can tranSMART have an impact here? This is a recent study coming from Nature Reviews Drug Discovery about lessons learned from AstraZeneca and looking at their drug pipeline. In essence, they compared trials that used molecular biomarkers as key readouts for trials versus those that didn't. And what they found is that 82% of projects that had an efficacy biomarker were successful in phase 2A compared to 30% of projects without a biomarker. So a dramatic increase in success rate. And in addition, 85% of all projects that they run now include a personalized healthcare strategy. And initial analysis shows that there's a 400% increase in success rate when you implement this personalized healthcare strategy using biomarkers. And what is a biomarker in this case? What does it do? It really ensures that we have the right target for the disease and we can differentiate that in terms of efficacy, that we're looking in the right tissue for bioavailability, for pharmacodynamics, etc. That we have the right safety, that we understand and know what the safety margins are, that we can clearly understand what is happening not only with the drug itself, but reactive metabolites, genotoxicity, and look at drug-drug interactions. That we're treating the right patients, the right patient with the right disease, so that the drug, in fact, can have its effect on that disease state. These are the key elements that are important in designing the right clinical trials to be able to move programs forward more effectively and to improve the benefits for patients. What is the translational research domain? It's really the convergence of drug discovery and clinical research, not into a single entity, but in fact into a communication in which the discovery process informs the clinical development process, and the clinical research informs the discovery process. So being able to collect all the data that we need, both from the discovery research side and the clinical side, to understand the complexity of the disease biology by collecting appropriate samples and bringing those together, linking the heterogeneous data types together, and being enabling of the analysis and visualization of those data. These are the key functions that we need to enable in bringing these together. When we do that, what we get, in fact, is a big data problem. That big data problem is not unlike the problem that Google has with their Google Earth and Google Maps. Is we have lots of different data from lots of different types of technologies, measuring lots of different things, measuring metabolomes and proteomes and microRNA and epigenomics and transcriptomes, SNPs, genome variation, full genome sequence. All this information that needs to be brought together to build an understanding of what is the nature of a disease state, what is the nature of drug action, who is the best patient to be treated with that compound. Well, in essence, all these data need to be centered, not like in a Google map where we have a GPS coordinate, but in fact by the patient, which is our GPS coordinate for healthcare. So we need to take all this data from all these different sources, from many different resources, and unify that on the patient so that we can now select appropriate cohorts, we can do our analysis, we can identify those biomarkers and those states that are necessary to push this forward. So translational research data is big data, not just in terms of volume, but also in terms of complexity. And the platform, the tranSMART platform, comes into this environment as a means to collect the data, to unify the data, to curate that data, to semantically integrate the data, so that we now have that organized in such a way that it can be analyzed and utilized. And not just by an individual scientist, but in fact by entire research teams, and those research teams being, in many cases, virtual and across organizations and across the globe. And what that data mart enables us to do by having that common data model, by having those semantic integration and the semantic regularization of those data, enables us to then implement a series of analytics across that that allows us to select the patients, analyze the data, and come to the conclusions and form hypotheses. So in essence, we can develop, in a hypothesis-free environment, new hypotheses about the biology of interest. So the tranSMART platform enables this, and the key for us in terms of collaboration is it's an open source platform. software is there for anyone to use. We have an open source contributing, and that community includes scientists, developers, service providers, clinicians. In fact, it's just a very diverse community of people working together in a collaborative way. And collaboration is really the key outcome of value from the tranSMART platform. The platform creates that value by linking discovery groups together with preclinical groups, together with clinical groups, enabling each of these groups to interrogate these data, to be able to form the hypotheses they need to drive their research, whether it might be a clinical trial design and bringing a new biomarker into that space, or understanding what is the appropriate target for drug discovery and choosing initial compounds to develop through that process. So tranSMART brings all these together and facilitates and enables that collaboration across these disciplines. And in our community, we've seen over the past 3 years a real broad adoption of the platform. Over 50 organizations have implemented tranSMART prior to version 1.2. These include international research initiatives like the ETRIX and EMIF initiatives as part of the Innovative Medicines Initiative in Europe, the TRAIT program, function of CTMN in the Netherlands. We've seen a strong adoption in pharma and biotech with companies like Sanofi, Millennium, Pfizer, Johnson & Johnson, and Roche making major investments in the platform internally. We've even seen government institutions like the FDA get involved with tranSMART. And then one of the things we really facilitated with the development of version 1.1, the first truly open source version of the platform, reliant then on the Postgres platform, has been the adoption by a series of nonprofits, including OneMind for Research, Orion Bionetworks working in MS, and the Critical Path Institute, amongst others. And this adoption has also been seen across hospitals and academia, certainly University of Michigan, Harvard with Paula Viek and others as well. And then we have a vibrant service provider community which has grown up to ensure that we can provide these platforms and support these platforms in the way that's most appropriate to each of the different groups using that platform. So overall, I think the platform has been growing since it was open sourced early in 2012. This is now the second major release of the platform, and the platform is an award winner. It won its second BioIT World Best Practice Award this past year for a project that was implemented on version 1.1 of the platform by Yanni Pandis, which is the UBioPred IMI project. And with 1.2 is having, in fact, almost exponential increase in capability and functionality. We expect these kinds of awards to continue in the future. I want to give you, just before we introduce each of the demonstrations, give you a quick view of the development activities that took place. Back in November, we had our foundation annual meeting hosted by Sanofi in Paris. And at this meeting, we developed a feature set for version 1.2. And that is, we looked at all the different features that different groups across the world had developed and decided which of those should make it into a 1.2 program. And then the foundation organized a series of hackathons, one in Boston, one in London, and then some testathons, that is to get developers along together with scientists to do testing. And those culminated in June of this year with a community testing release of 1.2. And then we did something unusual in the open source world, is we engaged the entire community in testing. And through June and July, we had 3 community testing debugging cycles where the developers worked very closely with the testers. And we worked through the areas of data loading and ETL development, functional testing and debugging, and implementation testing and debugging. That resulted August 1st in our first production release. And today we announce that formally and roll it out. And what you'll see is at the end of this week, in fact, is one of the first patches that will be formally released as well, as we continue to patch and update the platform to meet the needs of the community. I love looking back at some of the key metrics in the development process. This is a graphic taken from the Jira platform, which we use to manage the development process. And what you look at is when we had our annual meeting back here in November, you can see that there was a lull in activity until we initiated the first hackathon in February. And we see a burst of activity with the hackathon. We organized another hackathon with another burst of activity. And then as we went along to engage the community in testing, you can see, again, a large burst in activity both prior to the testing release and then post the testing release, to where we have a tremendous outpouring of effort both in terms of issues reported by the community and in terms of issues that have been resolved by the development community. One thing we can see is we still have a gap in our developer resource, and that's something we need to get more people engaged in to continue to enhance the quality and reliability of that platform. But it really shows the tremendous effort the community has put in place to put the platform together. I just want to take 2 minutes to run you through some of the key contributors. The community was very active across the board in evaluating the platform and reporting bugs for the developers to fix. And you can see the key contributors here. Terry Weymouth from University of Michigan, Jay Saha coming from IDBS in Europe, Ruslan from the Hive in the Netherlands, Jay Bergeron from Pfizer and Etrix, Tatiana coming from Rancho Biosciences, and others. We had a very broad distribution of people working both across the community and, in fact, globally overall. And then when it comes to development resources, developers that were engaging with these problems and fixing the problems, we see the same kind of global collaboration. Terry Weymouth from Michigan, Peter Rice from Imperial College of London, Charlotte from Sanofi, Axel from Imperial College, Ruslan from the Hive, Florian from Imperial, etc. So we can see the group of developers coming together to resolve these issues, in fact, span the community and span the globe. It really is a global resource and a global community that's come together to develop the platform. When we look at the overall contributors to this version of the platform, it really is quite broad. We have not only Etrix and IMI, CTMM, and others, but also groups like Converge Health, IDBS, BT Global Services that's hosted the platform, University of Michigan, which has provided key development and project management resources, etc. It's really been a global effort of people coming together to develop the platform that you're going to see momentarily. With that, what I'd like to do is turn this over to the key leaders here to show you the functionality that is present in this platform. And I'd like to ask Brian Athey, our CSO at the University of Michigan, to take over and make those introductions.
Unmuted.
Brian?
Oh, yeah, thanks. And I'm going to rush as quickly as I can, Keith, because I'm anxious to let everybody have a chance, the developers themselves and the key contributors have a chance to show their wares and keep us on time. Kevin is loading the PowerPoints now. What you'll see in a moment is the feature list that was prioritized at our Paris meeting back in November. And these were prioritized. It was a great team effort that led to this prioritized list. And you can see it's a mixed bag. Some of it had to do with types of data, like time series data, and the ability to incorporate dimensional data or take care of things like IDs. But some other features involved, bringing in and integrating key other analysis platforms like Galaxy or Cytoscape, or knowledge bases like Metacore. And so you can see that we had a prioritized list in Paris. Next slide. And this list, which lists the 1.2 release features of the analytics, we have separate data that will be released as well. We've more than satisfied all three of those priority elements that were identified in Paris. And the groups that have been contributing to this are, quite diverse, and we're going to hear from some of those groups today. So you can see our interfaces and faceted search, as well as these more analytic platform interfaces, and several items in between. And so, this has been a big challenge from the point of view of moving from 1.0 through 1.1, which was largely bringing on Postgres, to having a unified code base that can work both in Oracle and Postgres. And it brings together an enhanced library of capabilities in the analytics space, as well as data set contributions that we in the content committee will be working with the Nature Publishing Group and others to ensure that those are peer-reviewed and stamped with the Creative Commons license and all the things that we need to do to remain open and transparent. Next slide. Here you can see that we have in the 1.2 branch brought together the functionalities both on the Oracle platform and on the PostgreSQL platform. Thanks to Terry and Peter and your teams for helping with this. This turned out to be a much bigger job than anybody would have imagined. And you can see the list of code contributors to the 1.1 and 1.2. Okay, next. And so here's the high-level list. No need to repeat it much, other than to maybe state that with the faceted search capabilities and other focus on the search and user interface, that this complements the analytics and the data and the models. Next slide. Okay, and so here's kind of the list of enhancements, starting with search and user interface. And this has really improved quite a bit. And of course, it's so important because at once we need a simple interface, but it has to be quite precise in our ability to bring up the information that we need, not only the data, but the metadata and the associated capabilities that will link that data and metadata to the analytics. There is a deep set of data dictionaries and ontologies in the middleware layer that allow this functionality to be manifest. And this capability can also support the cross-trial and the save query to workspace functionality, which in the end of the day improves the processing and workflow capabilities of the tools significantly, as well as positions us for repeatability of running various workflows. Next slide. On the analytics side, here's our list. We will see several examples today. The Galaxy example will be shown by Peter and Giannis. The GWAS example will be by Jay Bergeron. The Dalliance Genome Browser was contributed by the Hive, and it was a very exciting contribution to have, and will provide quite a bit of value, especially with the emergence of even more ENCODE data coming online just this week. These are libraries and some of the open source capabilities that people have grown to expect in tranSMART and makes it special. This library has been improved in its functionality, as well as the ability to export data from those libraries. And we've already mentioned the time support and longitudinal study support, which is key in this era where we're not looking at just point source measurements anymore, but time series measurements. Next slide. Okay, and so here's some of the improved graphics and capabilities that some of which you'll see, some of which you've already seen or are already using, and some of the new workflows that are in place, especially against array CGH functionalities. And we're very excited that we're now supporting the RNA-seq and RefSeq and the other kinds of bioinformatics data that are replacing microarrays in many cases. Okay, data and data types and models. This first line is very important, and it's just an elaboration of what I just said. Going back to the slide that you saw where the integrated omics is the big data problem, that has to be parsed out in layers, really. And now we're able to look at proteomics data, microRNA data, and transcriptomic regulation data more generally, RNA-seq and gene expression data, the modern stuff, metabolomics, and VCF. This is really a great step forward for us. I've mentioned these other capabilities, so in the interest of time, I think we can move on, but you're going to see some of those shown. And so here's a list of the data sets and the contributors. And, we're moving beyond the GEO now. We're very excited to have the MAGIC data come online, some metabolomics and proteomics data from Sanofi and others, and microRNA and high-dimensional serial data and VCF data. This is a very important complement to the data that was largely geo-focused in the earlier releases. And as I say, there'll be more about this data set curation and appropriate release in the coming weeks and months, and we will be featuring this in our meeting in Ann Arbor mid-October. Okay. So this is obvious. It's a broad community effort. This is repetitious, so I think we can move on from here and get to the next slide, because I think we're close to getting into the demos. Okay, and so our first demo is going to be, Jeek, you are, I think, ready to go, and we're going to have you, show your demonstration of the search and browse function from Sanofi. And Sheikh Sui is working closely with Sherry Cow and others at Sanofi. And we're going to turn it over to you now to see the presentation of the enhanced browse and search functionality and look at some of the new data types that we're able to handle with the tranSMART 1.2 platform. And thank you, everybody. This has been a great community effort and a lot of progress. Thank you. Thanks.
Okay, so can I start now?
Yes, please, Jack. Go ahead.
Okay, yeah. So thanks, Brian, for the highlights. So I recorded a video due to the instability of the servers this morning. So I think it might be better to play the video. And anyone feel free to stop me anytime for any question. And so let me play the video right now. Okay, so the previous version of tranSMART can only hold processed subject level data for statistical analysis. And the new version can store metadata, for example, the information files related to a program, a study, an essay, or a analysis, making tranSMART a all-in-one depository. And let me hold it for a second. And the browse is the interface to view, manage, and search those metadata. The Analyze tab, formerly the Dataset Explorer, is now synchronized with the Browse tab, meaning that they share the same selected studies, which is extremely useful because after a study of interest is found, user can switch directly to the Analyze for the analysis of the study. And there are 5 types of objects in the browse. They are the program, study, essay, analysis, and files, and file folders. The file folders can reside at any level. And so this is the node of a program. Clicking the node displays the information related to this program. Title, description, associate text, and associate studies under the program. Right now, there's only one study under this program. And the power users can also have the ability to adding studies or adding folders under the program. Clicking the study node displays the related information for the study. And similarly, the power users have the ability for addition of analysis essays or folders. And there is a analysis node under this study node, and the users can add new folders or delete essays under this node. And so there is a folder under the essay node, and the folders can be a place to save files. Here there are 3 files, and users can add or delete files. Notice that right now in the export cart, there's only there was no okay. Let me go back a little bit. Okay. Notice that after user clicks the add to export, the files is added to the cart, and clicking the cart displays the file, and the user can download the file. So if there is a large number of studies, it might be difficult to find the study of interest. So the browse provided a search functionality. Now, if I type test, it would display all the nodes with the word in its name, or like in this node, in the description. Then users can use the filter to further narrow down the list. For example, if the user is interested in a cancer studies, notice that between the search categories, it's the AND relationship. But within the categories, it's the OR relationship, which can be changed to AND as well. And finally, the list can be further filtered to only one study. Now let's clear all the filters. There are different types of fields in the searching, except the free text, which searches the node's name, description, and the file content. All the other fields have the auto-complete capabilities. For example, if I want to search a gene, I will select the gene field and type ERBB, and it will auto-complete the item. So I search the ERBB2, and here are all the studies that with this gene as a biomarker. And by clicking Open in Analyze view, it switches directly to the Analyze tab. Notice that the filter persists through the Analyze tab. And that's the basic functionality of browse. We also enhanced the capability of tranSMART by developing the ability to take the new high-dimensional data types. The new data types are the MRI-seq, micro-RNA-seq from NGS and qPCR, and proteomics data from protein mass spectrometry and immunoassay from RBM, and also the metabolomics data, which are the quantities of metabolites. Now let's see an example using the RBM data. Let's switch to Analyze tab, and search RBM. And let's check the female as subset 1 and the male as subset 2. And under advanced workflow, let's choose hierarchical clustering. The clustering would be done on the RBM data of plasma. And let's click in the HDD button. Now let's use all the data. So if we do not input anything here, we would be using all the data. And now let's run. And it's taking a little while. So here is the heat map in two subsets. Yeah, now we see the heat map and hierarchical clustering on the RBM data across all the samples. So since all the high-dimensional data types works the same way with the advanced workflow, I'm now going to demo more of the high-dimensional data type. The next one is the serial data. Serial data is a series data on the same sample, maybe at different time points. It could be either low-dimensional data or high-dimensional data. In this new version of tranSMART, each node of the serial data can be associated to a numerical value, and the serial data can be plotted along the scaled x-axis using line graph. And let's see an example. Let's select the study test serial clinical data, which contains the low-dimensional clinical data at different time points. And let's use the line graph. And let's check the glucose level at different day points as the time concepts. Let's remove day 14 so we can illustrate the point that those time points will be scaled on the x-axis according to its numerical value. And let's check the trial arms to the group concept and then run. Now we see that the x-axis are the different time points, and the day 7 and the day 21. They are scaled according to their numerical value. And the y-axis is the glucose level. So the different colors are 3 different groups. The serial data also works with the on the high-dimensional data. Now let's drag this serial gene expression data into subset 1. And again, select line graph. And let's drag the baseline week 1 and week 2 as the time concepts. And let's select the gene ERBB2. And let's drag the sex as a group concept and run. And now we see that the x-axis are the different time points, and the y-axis is the expression of the ERBB2 gene in the two groups, male and female. If we want the whole data set to be in one group, we can clear the group concept and just run. And yeah, so now all the samples are in one group. So the next example is the heat map of high-dimensional serial data. Let's clear this one and drag the female as subset 1 and male as subset 2. And under advanced workflow, let's choose heat map. And let's drag only the baseline and week 2 into the field. And let's select all the genes in the apoptosis pathway. And let's aggregate all the probes. Yeah, so here is the heat map. We can see that the columns within each subset are ordered by the time points. And we can also specify to group by subject instead of the time points within each subset. Now we see that the columns are ordered by subject instead of time points. So besides the pros, new HDD and the serial data, we also made a significant improvement on the grid view. Grid view is a great tool to get a quick sense of the data, but the previous function was pretty limited. And in this new improved version, the grid view can display sample IDs, display categorical variables in a single column. It can hide or show columns at user's choice, and the user can also export to Excel at the selected columns or all the columns. So here is an example. Let's drag the diabetes to subset 1 and obese into subset 2. And let's run the summary statistics. And then let's view the grid view. And we can see the obese and diabetes, they are at different columns. And there is also a risk column that has nothing. Now if we drag the trial arm into the grid view, we can see that now the category variables, they are in the same column. And now we can delete those columns that we do not need. And we can also drag a numerical node into the grid view. Yeah, now we see that the height is in a grid view. So the whole table can be exported to Excel. And users can also select specific rows to export. Yeah, so I think it's a extremely useful feature to get a quick sense of the data. The last improvement is on data export. When I recorded this demo, the server did not work, so I did not demo this. But basically, instead of export the whole data set, now the user has the option to drag the individual node into the data export field and to export only those nodes. So finally, this improvement is a team effort, and at Sanofi, the team members are Claire, Anik, and Charlotte. In Genzyme, it's Sherry, Adam, and me. Our partners are Converge and also the Hive. Finally, I would like to thank the tranSMART Foundation for the great effort to make this community project possible. Okay.
That's great, Jack. Thank you very much.
There were actually quite a few features there. We've asked Dave John, who's up next, who will be showing the cross-study platform from Converge Health, to up the magnification on the screen so it's easier for us to see. I know you have some ability on site to make the mag a little bigger, perhaps sometimes, but if you could, Dave, if you could run it a little bigger, it'd be easier to see.
Sure. As soon as I start sharing, I'll check with everyone to make sure they can see.
Super. Thank you. And thank you, Jack. We really appreciate the hard work over there. There's a ton of features that we're going to be able to make use of there. There's no question about it.
Yeah, thank you. And sorry for this technical glitch.
We take risks and sometimes we make mistakes, but that's what it's all about. We're a team.
Yeah.
Okay, good. That's easier to see.
Okay, good. I just wanted to test that before I actually launch the presentation.
That's super.
Okay, so let me start off with kind of giving an overview of what I'll show. So I'm from Converge Health, but we were part of Recombinant prior and helped do a lot of the original development for tranSMART. But one of our most recent projects was with the FDA. And until now, when doing your analyses in tranSMART, you could only do one study at a time. So if you wanted to compare patients across studies for different reasons, you really didn't have a good way to do that unless you're just doing each analysis in one study at a time and then comparing the results. So what we enabled for the FDA was the ability to start to actually look at data across trials in one analysis. And what they wanted to do was, since they were getting device information and drug information and vaccine information for all sorts of different areas that would potentially treat the same disease. What they wanted to be able to do was compare treatment options to find out if they were more effective for than others for specific outcomes, or look at adverse events or safety issues and determine if there's drugs that actually are safer or result in fewer adverse events overall. They had a lot of focus actually on devices, but they also were focused on, like I said, vaccines and drugs as well. So we did this project with the FDA about a year and a half ago, and just to acknowledge who was part of the team, we had a team from Converge Health, mentioning everyone on the left there. And then as for the folks at the FDA, we engaged with the Centers for Device Drugs and for vaccinations, and CDRH, CBER, and CDR are those three centers. I couldn't remember everybody in the list, so at risk of forgetting somebody, I just wanted to acknowledge the centers themselves. So with that, what I'll do is I'll show you a little bit about what we've done. And I'm going to start off and show you an instance of tranSMART that is not 1.2, but it was the foundation for what we've done in 1.2. And the reason for that is because I just wasn't able to load enough studies into 1.2 to really show off the features. But I will show you how it works in the new version as well. So why don't I get started here, and I can show you, this is Dataset Explorer, now called the Analyze tab. And in my particular case, I'm going to look at asthma and COPD patients. So in this case, what I might want to do is just compare asthma and COPD patients across multiple studies. And to give you an idea of what studies are loaded, I can click on the studies. You can see I have a lot of breast cancer data, but I have 2 asthma studies and 2 COPD studies that are listed here. So it's going to look across both those studies when I do summary statistics. And I get the standard histograms back for age and for the pie charts for sex. And I'll also see race information. In this particular case, no race data was collected for these particular studies, so that's why that's not there. But then if I want to drag other information in, just like you would do in a tranSMART looking at a single study at a time, I can drag in something like smoking behavior and see how many patients were ex-smokers, non-smokers, or smokers in both sets of studies. So I'm looking at 4 studies simultaneously here. Now this is all well and good that I can get some idea of this information. And one thing to note when looking at this is I can see that, for instance, my second subset here, which are my COPD patients, they're a bit older than my overall population for asthma patients. So if I want to do a more in-depth analysis on this, and I can look at, I'll do a box plot in a moment, but I want to look at one of their lab findings associated with their disease, I'm going to limit the age to folks who are just over 45 to make sure that I'm comparing a similar population. So I'll come back to the comparison tab. I'll clear what I'm doing, and now I'll just have my population be patients who are over 45 years old. And I'll move into an advanced workflow. I'll start off with box plot. That's the one I'll be demoing today. And what I want to determine now is there a difference in one of these lab findings based on the condition that the patient is in, or what the diagnosis of the patient is. So I'm going to look at the different lab tests. I can see I've got forced expiratory volume, which is a good measure of lung capacity and lung health. And I want to look at that with the various trial arms and see any difference amongst these 4 studies that I've got here. So I'm going to drag all these trial arms in. And I'll just run this as is. And what that'll populate back is a box plot looking at all of these different areas for my forced expiratory volume. And you can see it's a little bit all over the map, as you might expect, but you can also see that my COPD patients have the lowest volume. So I can see this across all these different studies. Now to take this one step further, if I wanted to group these, I could actually rerun this analysis and use binning I can manually bin the groups. I'm going to manually bin the dependent variable, I believe. Excuse me, the independent variable, which is a categorical variable. And what I want to do is I'll put all my healthy controls into one. I'll put COPDs in maybe bin 4 over here. The folks that don't have COPD are also qualified as healthy controls. And then we've got maybe one non-severe asthma, and I think the rest of these are all considered, actually, I think one severe asthma, the rest of all these are considered non-severe. So then now I'm looking across multiple studies, and I run the same analysis, and I'll just come back with just 4 plots for the box plot, and I can see that there's definitely a trend associated with the categories I just described. So that's the idea behind what we enabled for cross-study analyses in boxplot. I'll show you now how that works in version 1.2. It's very, very similar to what I just showed. The only difference is it's only going to be going across one study, or because that's all we were able to load for test purposes. So if I look at 1.2, in the navigate terms section, instead of having across trials on the right, it's just below here. And I could drill in, just as you saw Jeek do a few minutes ago, with all the different areas. Now, for across trials, I'll drill here. And in this particular case, I'll actually substitute my population by those lab tests in this case. So what I can do now is look at forced expiratory volume, do that by a numeric value. I want to say something like 80 is a good number to divide up this group somewhere in the middle there. And then I'll just run the summary statistics the same way I did before, and I come back with all the same graphics. And this would be going across all the studies loaded within the across trials tab. I could also move in and go into box plot and do the same thing I just did. Actually, let me clear this first. So if I clear this and just bring in all of my patients, for instance, if I just bring in, if I look at interventions, for instance, or if I look at studies, maybe in this case, I think if I just look at one study, I can also bring in a second study if that were there. I think in this case, I actually only have data loaded in the first study that's shown here. But now I can do that same box plot that I just described, where I can bring in the various trial arms, look at forced expiratory volume, and run. And it comes back with a very similar graphic to what I just showed. Again, it's only limited by the data right now. But that was all I wanted to show today. So if there's any questions on that, let me know. I know it was a much shorter demo than what we just saw, but hopefully it makes sense and everyone understands it. Any questions?
Okay, super. Dave, that was great. We could see it well. It's really neat to see how some of these functionalities are being transferred from development work done at the FDA into the community more at large. And, it's also good to see how smoothly the thing is running with this new capability. Jay, are you up? Did you hear us?
Oh, yes.
Oh, hi.
Yes,
great. We're all excited to see what Pfizer is offering with regard to GWAS capabilities to the foundation. Sure, Pfizer has as good a tools as anybody, and it's really an exciting day for us to be able to show this.
All right, so let me do this. Okay, let me see, minimize that. Can everyone see my screen?
Looks great.
Okay, so I'm just going to run through a couple slides, and then I'll show you a quick demo. Okay, so basically the thing that Pfizer added into tranSMART is support for genome-wide association studies. I'm sure that most of you on the call understand what these are, but fundamentally, I've got the definition from Wikipedia. Basically, they look for allelic differences
between cohorts of subjects that exhibit a certain trait versus those who do not. And for example, it could be a disease-related trait. And fundamentally, one of the ways to visualize these is looking at a Manhattan plot, which is described here, where chromosome positions are on the x-axis and a negative log p-value, so the p-value of the difference essentially between the odds ratios for each cohort, for each individual variant. Typically these are single nucleotide polymorphisms, but that p-value is going to be on the y-axis. And in this case, you can see in chromosome 6, there looks like a region that has some substantial significance for particular SNPs for whatever cohorts were used in this experiment. So basically, a couple of years ago, our business groups came to us and said, you know, we're really interested in GWAS. We're using this type of genetic information to try to select our targets. And it would be great if we had a centralized system and a Manhattan plot viewer associated with that. So at the very least, we need a database. And actually, there's a product that I believe was created by Brian, probably your group, in University of Michigan called LocusZoom, which provides some of the functionality where basically you feed it the data and it'll come back with a Manhattan plot, say through email. And that was the minimal that we needed to produce. But we looked at our opportunity with tranSMART. We decided to enable this type of capability within tranSMART, and that would be good for our customer groups moving forward. So we worked with Converge, well then Recombinant. We leveraged the faceted search to provide a visualization and presentation of this data within tranSMART, and we built a thick client for the Manhattan plots and called the Genome-Wide Association Variant Analyzer, or GWAPA, and I will demo both of those today. Basically, what we're finding is that in general, people are telling us that they're saving weeks in terms of applying genetic information within the context of target selection. But at this point in time, we have over 500 individual GWAS analyses representing approximately 2.5 to 3 billion records in our database. Over 300 of these are imputed and have approximately 8 million SNPs associated with the summary statistics. They're used across many of our research units, and we also can represent expression quantitative trait loci, as well as metabolomic associations, although I won't demo those today, specifically demo those today. So I'm just going to end. There was a number of people who have been involved over the last couple of years, but I certainly want to give stand out to Converge Health, who did basically all of the programming for us. And on our business technology side, Peter Henstock was the person. He's one of our he's actually a very, very good programmer, but a statistician by training, and he actually built the Guava tool that you'll see. Ami Kandeshi, who recently left the company, but she was our technical lead for quite a while and worked with Terry to try to get this in, and really call out Haiyan Zhang and Hugo Berube. Hugo is our new technical lead who took over for Ami, and they've been instrumental to try to ensure that this was working for the foundation. Okay. So what I'll do first is I'm going to pull up tranSMART. This is the Oracle version of the Foundation's tranSMART, and what you're seeing here on the GWAS tab is essentially a view that on the left-hand side there's the faceted search filter, and on the right-hand side are the various GWAS analyses. So you can use the filter to get at the analysis that you'd like, and again, there's only a few here, but you can go in and search by a particular study. I'll pick this one, MAGIC, which is an open source, publicly available data set. So I'll select that. And if I go in there, I can see the MAGIC analyses, and I can further restrict either from the browser, or if I just click the ones that I might be interested in, fasting glucose and fasting insulin. I'm sorry, hit the wrong button. But if I pick these both, add to the selected filter, basically I'll see these analyses. If I open these up, what I'll see first and foremost, very, very quickly, is the top 500 most significant SNPs in any of these analyses. And you see that there's a number of values that can be associated with these GWAS sets. The GWAS sets are actually loaded through, we use a standard format, and there's actually a loader in this Upload Data tab. I won't demo that today, but basically you can load these sets directly from tranSMART. And unsurprisingly, basically what you see is, of course, individual SNPs, the p-value associated with them. And in this case, they are in order of most significant first. And we also have things like the gene, if they're in a gene region, whether they're in intron and exon, if that's the case, as well as the chromosome position. All right. And basically, if you want to see the Manhattan plot, you can click Manhattan plot. And because it takes a little time, I have this already set up. And what you're seeing here is Guava, which will launch directly from tranSMART. And actually, you can launch Guava on its own. Basically, it uses web services to get at the data that is within tranSMART. And I'll just give you a very, very basic demo. So if you look at, let's go back to tranSMART for a second. If you look at this fasting glucose analyses, you can see that this G6PC2, this gene was very highly significant. So basically what you can do with Guava is you can select your available models, in this case the magic models, and you can give it either a list of genes or RSIDs, and you click Retrieve Data. When the data comes back, as it is done for this G6PC2, what you can do is you can see the results, in this case on a color plot. So the right-hand panel here will list all the analyses that are retrieved. You can click on this, click the color plot, and what you'll see is this region where around this G6PC2, in this case, you can select the flanking region within the query, and you can modify that. By default, it's 100,000 base pairs. And basically, you can mouse over the SNPs, and you can get further information on any one of these. And this was a meta-analysis, so for the folks who are seeing these very, very significant p-values, it's because it's a meta-analysis. And you can do some other basic things, such as you can zoom in to regions, of course, zoom back out, and we'll go back to the original. Additionally, you can do things like add lines. So if you had an interest of saying, I'll call it my line, and I'll put in a p-value, a negative log p-value of 50, and it'll put in lines so that you can easily see, and little tricks like that that make this relatively easy to use. You can also put in shading for gene regions. Additionally, although there's not any in this position, we also have loaded recombination rate, and where it's available for positions within the genome, you'll see the recombination rate as well. All right, you can also view this as a thumbnail plot, and what's interesting is unfortunately I don't have more than one model selected, but basically you can add in multiples of multiple analyses if they're on the same genome region into this interactive Manhattan plot. So you can see multiples and you'll see color differentiation. You can also view these as in a thumbnail format. Basically, if you're looking across or looking at varied regions in the chromosome, you can present these and it will normalize all of the plots on the y-axis. So you can look across, and you can do things like resize, and you can get put a number of these plots together, and again normalized on the y-axis. The last thing that I'll show you that you can do that I think is interesting in terms of just being able to get at the data in a tabular format. Go to a table view, and right now I'll remove everything. And basically, I can select a model of interest and include all of the data. So here's all the data that's associated with that particular model. I can, for example, maybe pick a p-value. Say I'll go a p-value greater than negative log p-value greater than, say, 10. I can remove everything else. Actually, I'm going to up it a little bit. Again, this is a meta-analysis. I'll say p-value 50, negative log p-value 50. And I can very easily go in and restrict these data and be able to export that out into the limited files. But it's a nice, easy way when you have to be able to get at these data quickly and subset these data very, very quickly, especially if you're viewing multiple of these models. So I think I'm going to end there and see if there may be any questions, but just a little taste of what you can do with the GWAS capabilities within tranSMART 1.2. Thank you very much.
Okay, I'm unmuted now. Hey, Jay, this is fantastic. We see immediate use for this. Do you identify variants and output in the VCF format?
So right now we don't. That's actually a great requirement. Right now we only export out in a delimited format, so a CSV format.
Okay, yeah, that'll be something maybe we can put up for further work. I mean, I'm very excited about using this tool here in Michigan, and I'm sure it's going to see broad use throughout the community, and it's going to be a big highlight for us. And seeing how we can integrate this with other tools, we've got a nice pipeline we've developed where this GWAS would be part of it, and we'd be happy to share that capability with the community at the upcoming international meeting in mid-October. Thanks so much for this. This is a great contribution.
Okay, thank you.
Fun to see Eric Bauman's name on there. I hadn't seen that for a long time. He, of course, used to work here.
Oh, excellent.
Okay, Peter, you're up next. This is a monumental, capability of bringing Galaxy in and integrating one platform with the other, and we're very excited to see your description of the demo that you made earlier today. And Giannis,
okay, thanks very much.
Yanni, are you online as well?
Yeah.
I think he is.
Yeah.
Can you see my screen?
We can, and we can see it, and it's big enough.
Should be showing Galaxy.
Yep.
Hang on,
Good.
let me just. Okay, how's that?
Perfect.
So this is Galaxy. Just to introduce what Galaxy is, it's a tool developed at Penn State that allows you to put all kinds of big data files in and do all kinds of analysis. So there are some data import functions here. There are ways to do various feature conversions. It can cope with a very wide variety of data types and data formats. Run statistics. It's good for running NGS analysis and various other things. So what we're going to do here is we're going to go into tranSMART and we're going to extract some data from tranSMART and pass it straight into Galaxy in a format where Galaxy can then be used to launch all kinds of analyses. So within Galaxy you can then build workflows that will take your data and run them through standard workflows that you have set up using the tools available. We can go to the shared data area, which is where we'll get the import from, and data will appear in this history over here, where we'll be able to see the data sets we've brought in and select them in Galaxy. So I'll just go into tranSMART. This is actually very easy to do. He says confidently, it worked for me this morning. So we go in, pick some data, we go into data export, we select the data that we want to export, and we'll just run that in the background. Okay, we've got some data that we can open up. All right. There we go. My version of WinRAR's a bit out of date. So we can go in and look at the data as usual in tranSMART. But that's not so interesting. What we want to do is do something useful with it. So we can go now to the Galaxy Export tab, and we take that data that we've just exported from tranSMART, and we now export that into Galaxy. So I'm running as Axel here, so let me pick a name for this that's appropriate to Michigan. There we go. And if I now go to my Galaxy tab, just a word of warning, there is some setup involved in getting this working. So I'm running as Axel in tranSMART, and I'm running as Axel in Galaxy, and there's some setup involved in linking those two so that we get Axel's data here and not anyone else's. So here we go. I've now got a data set that's appeared in Galaxy, and I can expand that in Galaxy. We've got the same subsets, the same data available, and I can click these files. I can view them here, and I can import them into Galaxy. Then if I go to the Analyze Data in Galaxy, they now appear as files 1 and 2 in Galaxy. I can now pick any of these tools, anything from the Galaxy tool shed. This is just the very basic set that we've got installed on the test server. There are hundreds of tools available in Galaxy, so this could be extended, exporting all kinds of data and running various analyses. So I can go in and view the data here, and then you have the full power of Galaxy available to you. And it just builds on whatever you can export from tranSMART. So that's the basic functionality. We're using this Galaxy Export tab here. Having exported data, we then send the data to Galaxy. The same as you'd have a zip file exported for your own use. You can export the zip file to Galaxy and run analyses there. Later on, I think we have a demo from Kees van Bokhoff showing the opposite, showing a utility that can then get the data from tranSMART. So we can do both. We can send data to other packages, and we can get data from tranSMART. That, I think, is what I wanted to show.
Right. Okay. Great. You know, we're looking forward to seeing some of the functionalities that we can run kind of back and forth. I guess my question, Peter, is that this is a bidirectional capability so that we can import data from the Galaxy output back into tranSMART. Is that part of it?
At the moment, no, but as we'll see from Casey's presentation, it's possible to get data, possible to talk to tranSMART from outside as well.
Yeah,
so
well, that'd be good future direction. So
what you could do, of course, is save the data and then load it in. So Galaxy would transform it into various forms that we could then bring it in. One thing we'd like to look at in Etrix as well is the capability to use analysis results.
Right, so I think what we'll look at is
for other things. It'll be a big area for us in the coming year.
Yeah, good. Yeah, I think so. And we can feature that in some of the advanced workflow work that we want to discuss at the Ann Arbor meeting. But this is an excellent first step, and I think it's a nice segue case to your presentation where you're going to show, and you know, we're very interested in becoming a fast user of the Dalian's genome browser capability, and you'll also show the RESTful API and you know, launching our capabilities with the RESTful API. So there's really, I think, 2 demonstrations that you're going to show. So I'm glad you're here, and you know, we're going to turn that over to you, Case.
Okay, so can you hear me?
Hear you great. Just like you're here, even though where are you today? You're in Italy, I heard.
I'm actually in Utrecht now.
Okay.
I planned to be in Italy, but something got in the way.
Yeah, work probably.
Okay, so can you see the slides all right?
We can see it fine.
Great. So the first demo is the genome browser. Funding for this has been provided by Trade, and we've developed it together with our partners in Trade, Philips Research, Erasmus MC, the Netherlands Cancer Institute, and the Vrije Universiteit Amsterdam. The idea here is that we wanted to be able to visualize genomic data in tranSMART and then be able to compare genomic variants between different patient cohorts. Also bring in copy number variation data and then compare that with background information that you can get from public service, for example, in code. So because we wanted to have a genome browser that is in place in the GUI of tranSMART, we wanted to have something that would basically use JavaScript or HTML5 to show it right in the user interface. We wanted to be able to drag in high-dimensional data nodes. And as I said before, genomic variants and copy number variation data. I can imagine that in the future we can also, for example, show protein data, but we don't have that yet. We try to use as much as possible open source software. Actually, one of the core principles of the Hive is trying to reuse as much as you can. And in this case, we were able to reuse Adalian's genome browser, which is developed by Welcome to Sanger, Thomas Don. We even had Thomas come over to a hackathon in Netherlands that we organized with Trade, which was really nice. And he helped us a lot to bring Adalians into tranSMART. The other component that we brought in is MIDAS, which is another open source software developed mainly by the European Bioinformatics Institute, and which delivers the backend services of showing the genome track data. Both of them are implemented as tranSMART plugins, and the tranSMART core API was also touched. I'll come back to that later when I talk about the RESTful API. So I'm trying to also showcase a little bit the technical background here, not too much, hopefully. So basically, we have the core DB, which is an implementation of the core API of tranSMART. And then in this case, we use the MIDAS plugin, really, as I said, to bring in the track information that you see in the front end. So as for a demonstration of the feature itself, just check. Yeah, okay. So tranSMART 1.2 server, as you've seen before in other demos. We brought in some cell line data, which are contributed by various members within CGMM Trade. And one of the assays we have on this data is a array CGH, basically copy number variation array data. So what I wanted to do now is show this on the genome browser. And all I have to do is drag and drop the high dimensional data there. If it supports the genomic track display, then it will automatically load the tracks in. So you can see here now that I have the losses and gains of genomic regions. If I would be able to zoom out further, then you could see also that the different genomic variations have, of course, different ratios of losses and gains, but I wasn't able to get the demo video in time with that, so just have to leave me on that one. The second is I also can show genomic variants on the browser, which allows me to look in a certain patient cohort, what are the variants in there. So I drag in that node again. What you see here is basically a visualization of the different variants. The pluses here are single nucleotide polymorphism SNPs. I also have insertions, the green ones are deletions, the red arrows, and other features which are hard to categorize and labeled as diverse, like this one that has a mutation that goes from ATAG to AAA. You can click on a variant to bring in more information. Like here you can see this is a deletion where the T is removed in the alternative allele. And I can bring in even my own attributes as demonstrated here by the testing. So one of the nice things that is a little bit hard to see here because I just have two if you shift data from 2 patients, but I can actually see what is the minor allele frequency, so the second most common allele, basically, what that allele is and what the frequency is, and that's also shown in the graph here. And with that, I can actually compare my cohort that I've selected with the frequency in the whole study. I have other information like the quality of depth. If I have that loaded in my VCF data, I would also get that here. Of course, I'm also able to navigate, zooming in and out, and I'm able to go to specific genes as well. The scrolling hopefully now should even work on an iPad. It's one of the things we try to fix. So going to a gene is as simple as just typing in the gene name. If we take, for example, tumor suppressor gene, TP53, you can see here the different variations that I have in that particular gene. And here, okay, so here you can see actually there is a minor allele frequency of half, meaning that we have the alternative allele in one of the 2 samples. Right, so that's it for the genome browser. I guess the idea is that I continue right on with the next one. Okay, so that's the RESTful API. So let me first mention the funders again. So that's Janssen. They funded this development that we did by their in-kind contribution to eTrix, basically. And I'm very excited of this development because what I think is that for tranSMART, if it really wants to become a leading platform for translation research installed at pharma companies, at academic medical centers, at nonprofits, disease foundations, etc., and if it wants to be a de facto data sharing tool, what you need for that is basically first the sound architecture of the whole platform. We also need options, lots of options for enterprise integration, like being able to plug in your own clinical data warehouse or your in-house omics data infrastructure. And you need to be able to connect downstream analysis tools. We'll give some examples of that. And hopefully, with these developments, we can not only have developers developing against the core of tranSMART and putting new features directly in it, but also connecting outside tools like great new visualizations or sophisticated omics analysis tools, which just would use tranSMART as a data repository. So I'm very happy that Janssen funded this coding of the RESTful API. Maybe to quickly highlight the various APIs that we have, the core API is a Groovy API, which is really, that's the innermost part of tranSMART, and it defines the language in which the components talk to each other. tranSMART Core DB I showed before. This is a reference implementation of the core API, and we have Oracle Postgres support at this moment. Though likely if you plug in MS SQL, it will work as well, because we're using Hibernate. And then this new development, which I want to show you, is the RESTful API. The nice thing of the RESTful API is you don't need to know anything about tranSMART, the architecture, how it works, to develop against it. The only thing you need is the API contract, which you can find in the GitHub. So quickly, from a technical perspective, what is the RESTful API? It only uses get in a moment, so it allows you to get data from tranSMART, both low and high dimensional data. It uses OAuth 2 as a protocol for authentication. This is the same protocol that, for example, Google, Facebook uses for their APIs. ProtoF, we use that for transferring the high dimensional data. As you can imagine, if you want to transfer microarray data over the internet, it's not a very good idea to encode it in JSON, so we use a compressed protocol for that, buffered. And like I said before, this thing, this RESTful API, allows you to build your own tools on top of tranSMART. And we have already 2 implementations, one for R and one for Spotfire that we built together with PerkinElmer, and they're actually selling this, I think, at the moment. But what I want to show you now is the R client. This is an open source R library, which just works against the RESTful API. So here's my R interface. I'm running R on my laptop. And first thing I have to do is load in the library that implements the RESTful client and allows me to talk to tranSMART. Next thing is I have to connect to tranSMART and identify myself so that tranSMART knows who I am and that I can actually get data into this R session. I do that by copying the URL that R generates for me here in the browser. That browser then gives me the login for tranSMART. So I log in with my account, in this case, admin account. And what it gives me back then is a code which can copy paste back into R. So in the background, the OAuth protocol is used, and what happens here is that tranSMART now knows that I am an authenticated user of tranSMART. So the next thing I can do is get all the studies that I have access to. I can do that by running get studies. So as you can see, I get back an object in R which represents the names and the different concept paths of the studies. And next thing, I can, for example, get all the subjects of the study. So let me just first show you the study as you can see it in tranSMART. It's a GeoStudy, which has mRNA data on 55 subjects. I can get the subjects by entering get subjects. And it just gives me all the clinical information that I have with the concept paths as, well, actually the truncated concept paths, so the concepts itself as the column headers and the data in the rows, of course. So with that, I can now actually also get the observations for a specific concept. Using data, I can then go ahead and use the powerful plotting abilities of R to basically make any plot that I want with this data. In this case, I'm plotting the FEV ratio of females versus males. As I said before, I can also use the RESTful API to get high-dimensional data, and R has an implementation for protobuf. Luckily, we didn't have to write that one. So next thing I'll show you is I have some RNA data here in the study, which is called lung. I'm using that name of the concept and matching that with content.match. And with that, I can get the actual microarray data. I choose as projection the log transformed intensity values of the mRNA groups. I can specify the word projection. I could also get disease scores if I wanted to, or the raw values. Then next thing is I can use, again, the R plotting functionality to create a heat map of this data. To speed up the data loading, I've actually truncated the mRNA data in the database to only have 55 probes, which then gives me a nice block heat map of the probes versus the patients. Okay, that's it for the RESTful API and the subsequent R client that implements it.
Thanks, Kees.
Yeah.
Can you hear me? Thank you very much for showing both of those capabilities. They're both going to become in handy, and I couldn't agree with you more that having the RESTful API is really going to be a key to having good, solid enterprise implementations. Your contributions to the tranSMART Foundation have been many and have been well appreciated. We treasure having you as part of the community. I want to thank everybody for the demos. As you can see, we had a chance to take a look at a wide range of new capabilities in tranSMART. It's still early days. Proof is in the pudding. We've got to use this stuff, get our institutions and others to adopt it. But I think all of you have put us in such a good position to do so that, if we look back and see where we were and where we are now, the trajectory is clear. We're moving up, and this has been a fantastic day for us. The next on the agenda is related to our documentation and video projects, to be followed by tranSMART rebranding, which includes not only logos, but the new website. And so we'll turn it over to Rudy.
Thank you, Brian. Hopefully you can hear me and see my screen.
Very nicely.
Yeah, good.
Okay, so we are going to, obviously getting code and getting all these great features built is very important. Of course, the next step is to get people to use it and really expand the community who's actually making use of all these great tools that we're building and the great platform that we have here. And so we have a couple of projects underway that I wanted to just give you a very brief introduction to in terms of building our documentation, sets of tutorials and training for the new version of the platform. And that's what I'm going to talk about very quickly here. Although I just wonder how many of you realize 1.1 did have some pretty good documentation with some very hard work at the University of Michigan and others who brought together a set of manuals and 8 tutorials. These are all on the website and have been there, and their tutorials are loaded and are very good, actually, to give you an introduction to at least a few of the features of the current system. And we even have, thanks to the Michigan team, a brief description of the differences in the interface between version 1.1 and 1.2. All of these can be seen on our YouTube channel, and I encourage you to take a look at that. But we are putting together, we have the teams now working with the developers and the teams that we've heard from this morning, this afternoon, and we brought in Eric Kaplan, who has joined us as the project manager for the documentation program. And the goals that we set forth is to build up a 1.2 version of the user guide, online help, a technical guide with a little more detail, the release notes, migration guidelines, and data loading manual, and we're working to put these all together. Again, as a community effort, this is going to be a lot of activities to pull together, but Eric is a technical writer. He knows and appreciates the science and a lot of what's gone into this, and we'll work to pull these together. But again, we're working to bring this all into a usable form, especially to help bring new people to use the platform and get the most out of it. We'll be rolling these out as they're finished, and you can watch and you'll see announcements and availability on the website. And we're hoping to have at least initial versions of all, if not most of these, if not all of these, by the annual meeting in 5 weeks. And so if you may hear from Eric, if you're a developer or working with the system or doing some testing as he tries to gather the information that he needs. Similarly, Alex Papa has joined the project. She is leading the collection and creation of the video and tutorial program. Similarly, trying to pull together a broader overview of what 1.2 is, differences in 1.1 and 1.2, and looking at the key features between, that are some of which we saw today and some of the other pieces that are there. And again, we're targeting the completion of this for the annual meeting to at least have first versions of all of these for everyone to be able to use. I wanted to highlight that, we're really trying to focus less on which buttons to push and really trying to get at what's the real goal of the various features and capabilities, what's the scientific use and value of them, and really have some good stories that we can really explain how the functions work, but also giving you some sort of tips on how what these were designed to do and what types of science you can actually do and what types of questions and answers you can ask. As I mentioned, we have YouTube channel set up now. This just shows the playlist for the tutorials that are already out there, again, from the University of Michigan. These are very nicely done, and you know, they are, you know, description of, you know, what a different feature might be, say creating a heat map and a full video of how you do it. The creating gene signatures, pretty, you know, might be, you know, a little boring kind of a thing, is because I just pushing which buttons to push, but they've done a really nice job in terms of capturing, you know, what sorts of buttons to push and some annotations to it. And then the advanced analysis tool, they even, you know, we have closed captioning in there to help you make sure you get a good picture of what the features are. What we're trying to strive for is to bring together a good script with the data set involved and all these brought together to create the tutorial, with the idea being that you can use these tutorials in a fashion where you can actually, you know, execute the same tutorial on your machine with a live data set and the live platform running on a demo site, so that you really have a chance to really use this, you know, each of these capabilities, and really learn what they are and how they function. And that's really the goal of, you know, pulling these together. How can you help? If you have been using the platform, you have some good scientific stories and some interesting data sets, we'd love to hear from you. Get an idea of what, you know, what your story is, you know, why you used it, what's the value, and you know, this team, you know, between Alex and Eric, they will pull them together and get them, you know, packaged up so that people can, you know, we can share them with the rest of the community. The third piece of this is a training program. We really have a goal to put together a set of courses. These at least will encompass a user training course, an admin training course, and we will get to an advanced training course, also with the opportunity potentially of having custom courses developed for specific groups. The goal is to have on-site training so that we can come to your organization, your company, or your university and teach a course. This could be just people from the organization, or it could be regional, you know, say in Boston or Cambridge or, you know, San Francisco, something where we'll come in and do a training. We're also looking at, you know, having a webinar-based training program. We hope to. The plan is that these are going to be very well done courses, with including, you know, manuals and good descriptions and course syllabi. So we're developing these. These will be fee-based, actually support the creation of these training programs. And these are collaborative efforts that we'll be doing with some of the tranSMART vendors who do training already, and working with the foundation to try to put these classes out there so that, again, our focus can be to expand the community and the number of people actually can use the program. And the platform. The first training course, we've been working with Rancho Bioscience to put together an introductory course that we are trying to organize to get at the annual meeting, and we'll hear more about this shortly. And if you are interested, though, please let me know, and we can talk about that. So that's the brief description of these three programs. Again, trying to pull this together for everyone. I'm going to spend just another minute just to talk quickly about our rebranding that we've been doing on the foundation, and trying to pull together, you know, just to bump up, you know, our look and feel of what we're doing. It's been 18 months since the foundation itself was founded. We've now had our second, you know, release of the platform. We have 18 member communities as part of the family, and 50 different organizations at least using the platform. And so, you know, as we're coming up to our second annual meeting, we thought it would be good to have a new look and feel for, you know, some of our capabilities. Now, I want to emphasize we have the same goals, the same mission. It's just we now have an improved platform, and we thought, you know, a fresh look, you know, might be kind of cool for us. So we designed a new logo for the foundation, which you see here, trying to make it, you know, the open, you know, in line with some of the open communities out there, and in terms of how the logo looks and the feel of it. A new logo for the platform itself. Again, we can use these to help us brand, you know, who we are and what we're doing. We've put together a new website. I'm not going to go through it now for time, but please go visit the website. It's been all redesigned. All the old information is there, and we've got a lot of new stuff that we're putting in. We plan to keep this, you know, fully up to date and hopefully pretty dynamic, so that you can get all the information that you need about what we're doing, the platform, the community, and the membership. and a blog is part of this, and so we'll have this available for everyone to look at. It's up there as of today. It is live. I'd love to get your feedback on it. We have started a newsletter, you may have noticed, as we announced this launch event, and so we will be sending out, instead of sending you 1,000 individual emails, we thought that we were feeling bad about doing that so much even during the summer. We're trying to gather all the information that we have and put it into a newsletter form so that you can go to one place and see what's upcoming, what some of the things that we're doing, and do that in an email blast for you. And so you should have seen this already. If you're not on the mailing list, please let us know. We can make sure you get on it. And then the last piece is, broadening our visibility in terms of presence at conferences, doing presentations, and really building ourselves a bibliography of what we're doing. And so, we've got a publication section on the website now where we're trying to gather up all the different articles that are published about, either about tranSMART itself or about people doing science using the system, which is even more important. And on the site, you have a chance, if you have published an article, if you've read an article that you think is interesting for the community, you can just fill in as much as you know about it. We'll track it down and get it added to the publication list. The same for presentations, the same for information that might be useful for the blog. So, again, this is our community, and so your participation is really important to help us really pull this together. As I said, we've built a YouTube channel, and so we've got on the YouTube channel our tutorials, but also the community webinars. All the webinars are recorded. They're out there for 2014. The Paris meeting, a lot of those are out there already, so we're pulling all these together and making these available through our YouTube channel. We have the LinkedIn group on LinkedIn, the tranSMART group on LinkedIn, and so here again, we're getting much more active here and trying to keep that up to date and giving you information if LinkedIn is your social media of choice, or if you like to tweet. We also have our Twitter account. We've again been trying to push a lot of things out there. We've got quite a few followers on both LinkedIn and Twitter, and so we'll continue to try to drive these as we keep you informed about what's happening and what's going on across the foundation. That's what I wanted to say, and I will pass this back over, I think, to Keith.
Keith, yeah. Thank you, Rudy.
That's great, Rudy. Thank you for that. Let's see here, going back. So I think looking at the agenda, we want to have a little time for questions and answers and go through that. As I look at the questions on the webinar on the dashboard, I see that there's a few questions that we can get to, and then I'd like to kind of remind people to go forward. Let's see here. Is it possible to have the name of the point of contact at the FDA who's implemented tranSMART? I think that was
that's Nancy.
Dave John. Dave.
Sarah's on there.
Dave,
yeah, I need to get back and check on that. They were hesitant just to give out the information, but let me check with my contacts and get back to you.
Okay. I think it's Sarah that is interested in that information.
Yeah, there are at least 5 offices.
This, yeah.
Yep.
And
Gil, I'm glad you're there. I just wanted to make sure we introduce Gil Oman, who's our chairman of the board, who's joined us today as well. And in my closing remarks, I'm going to tell you a little bit about the structure of the foundation and things going forward. But Gil, if you want to say hi there for a second, that'd be great.
Yeah, greatly appreciate everybody's efforts leading up to today. It's a big day today and tomorrow morning. So let's go forth and have a great meeting in October.
Fantastic. Let's see. I think, Gil, you had a question for Kays. He said, would you briefly highlight the advantages and limitations compared with the direct use of the UC Santa Cruz browser? Kays, are you still online? Can you help us with that?
Sure, I can take that. I guess the biggest advantage of using the browser in tranSMART is that you can drag your tranSMART data into it, which is something you can do with the US Sept browser. So does that answer your question, Jill?
Yeah, that's really the key point, I think.
Case, is
there may be some other features that you want to mention that might be under development for Dalliance. in order to be competitive with what UC Santa Cruz permits people to do for analysis and visualization.
Right, like methylome analysis or showing histone marks or uploading ENCODE data and having that available to compare to what is being analyzed within tranSMART. Is there any plan along those lines?
Well, a browser is of course just a browser, but the key thing is the data. And then, at least in my opinion, the nice thing of the browser that we use is that it does implement the DAS protocol. So any track like the ENCODE data that is served on a public server, whether that's from the EBI or from UCSC, which supports this protocol, can basically be visualized in the genome browser. I think that's so it doesn't have to be this or that. You can actually combine and mix those.
Boy, that's super. We're going to give that a try, and we'll report back to the group on how we do.
Yeah, thanks for clarifying.
That's something we want to do, Terry.
Yeah, and maybe what's good to add is one of the features that I'm very excited of, which is going to come, and that we're developing in trade, is bringing the OncoPrint visualization from cBioBortal to tranSMART. So that's another example of basically collaboration that's we're going to see later.
Great. Case, I see another question that you can help us with. Dave Merberg has asked, in the demo, an OAuth key was retrieved with a web browser. Can this be done programmatically instead? And that's for the R interface.
Well, that's a good question. I think that's possible, as long as, of course, the thing is then you have to write that. But yes, sure, you can automate that. Basically, you have to submit your credentials then programmatically to the website, and it would retrieve it. Sure, that's possible.
Okay, great. Are there any other questions that we have from the audience? Anyone want to raise a hand and ask a question verbally? I don't see anything else. Okay. Brian and Kevin, are there any questions in there at Ann Arbor that want to be asked?
Guys, Jerry, anybody? Questions? No. No, I think I think it's been pretty straightforward. There's been a lot of information. Obviously, a lot of the group here has been very active all along as part of the team bringing this all together. But it's exciting, and I think over the next weeks leading up to the meeting, we should continue to try to work with these things as we're going to do here locally and with our partners, say at Johns Hopkins, and bring issues and items for version 1.3, which will be a discussion amongst the code community and content committee. We want to bring that together so that we can see what is really in the near-term horizon. We've got some interest in bringing imaging capabilities and integrating the capabilities from the Laboratory of Neuroimaging, LONI, at USC. That's going to be part of a hackathon. Bringing in WACA, machine learning, workflow capabilities, tools from and data from the Institute for Systems Biology. So there's a big future ahead. So this is a great place to rebrand, relaunch, and grow the community, which I know is central to Keith's vision for growing the tranSMART Foundation over the next 18-month period.
Great. Thanks, Brian. What I'd like to do is kind of circle back to something we did earlier, just really quickly as we close, which is to take a look at this is one view of the effort of the community working together. And I think this really highlights how the foundation acts as a catalyst to bring the community together, to work together, and seeing how much work got done, particularly in the last 6 months, from the initial hackathons back in February through the production release in August, and see how everyone really brought things together. It's just been, from my perspective, working in this community has been fantastic. The efforts of Kevin and Terry at University of Michigan to help organize, Peter, Yanni, the whole team at Imperial College, UK's group, et cetera. But it's been an amazing effort of people, both individually and collectively contributing. And as I said, it's been a fantastic collection of groups that have come together to do this. But the foundation is a member-driven program, and the resources that we've been able to pull together here are driven today 100% by our membership program. And I want to let people know we're kicking off the second iteration of our membership program. We want to encourage, we have 18 member organizations and over 50 different organizations using the platform. To me, that's an opportunity for more than 30 groups to join the foundation. And we'd like to get you to join at some level going forward. As we said, we have gold, silver, and bronze memberships. And the structure of the foundation, Gil being here as our chairman of the board can tell you about this, but the structure is a management team that is directed by a really engaged board of directors, which is led by Gil Oman at the University of Michigan. We have two key committees of the board, which are our governance and nominating committee, led by Matteo Di Tommaso at Pfizer, and our finance audit committee, led by Brett Davis at Converge Health by Deloitte, and just a very engaged group. We have our 3C committees, which are really engaged in the three aspects that we see driving forward in this community, and that's code, around architecture and coding, etc., content, the data that we share together, and community, how we all come together to work collaboratively. So this is our organization as we go forward. We do have today 6 gold members: Deloitte, Oracle, Pfizer, Roche, Sanofi, and Takeda. We'd like to grow that representation as we go forward. We have 2 silver members: Johns Hopkins, University of Michigan, and 7 bronze members: AssureRx, Cognizant, ISB, OneMind for Research, Sama Technologies, The Hive, and Arlion. Our goal is to really more than double this effort and to get more representation across the community on our board of directors. Where that implementation of community comes together is at the board. And this is the board of directors that we have today. As I said, we're led by Gil Oman, our chairman. And then we have from the gold membership, Jamie Dannenberg from Takeda, Brett Davis from Converge Health, Matteo Di Tommaso from Pfizer, Olivier Gien from Sanofi, Jürgen Hammer from Roche, and Jonathan Sheldon from Oracle. We also have, besides Gil representing the academic institution, University of Michigan, we have Ken Pienta from Johns Hopkins University. At the bronze level, we're represented by Jim Burns at AssureRx and Lee Hood from the Institute for Systems Biology. And then as we balance out our board, we bring in members from around the community. We have Hiroki Kitano from the Institute for Systems Biology in Japan, Anne-Marie Martin from IMI, the Institute Innovative Medicines Initiative, Gerrit Meyer from Trait, and John Wilbanks, who's a key fellow with Faster Cures in the open consent space. So this is the board as we go forward, and we'd like to see more members joining that board and to continue to grow it. In addition, the way to get involved is to get involved in our 3C committees, the code, community, and content. We'll be coming together again in person at our annual meeting coming up in October in Ann Arbor, and working with the various 3C committees, spending half a day working together on various initiatives there. Remember, your leaders for your code committee are Ike Guo, our CTO, and Jay Bergeron from Pfizer. The leaders for our content committee are Brian Athey, our CSO, and Cyril Osheron from Thomson Reuters. And our community committee is Kevin Smith from University of Michigan and the foundation, working with Sherry Kyle from Sanofi Genzyme. And these groups are accountable to the board and help direct the management team to make sure we're doing what the community sees to be most important. As I mentioned, I would really like to encourage those of you who are at companies who have not yet become members to find a level of membership that works for you and to join the foundation. It's your involvement, it's your membership that really fuels what the foundation is doing for you. If you need a membership package, which consists of an application, bylaws, etc., contact Kevin Smith at the foundation, and there's a link on the website for getting there. But I'd like to finish with recircling back 18 months ago when we set out to build this foundation together under Brian and Michael's leadership. Is we set up a 3-phase plan in place to deliver. The first was to develop and deliver version 1.1, which we did last year. was to establish an operations team that could really grow and build the foundation, and to initiate a membership program. We accomplished all those last year. Our second phase was really around formalizing and stabilizing all our operations, which we've done quite well, to deliver version 1.2 and to convene our member-driven board, which we've done. We still have as goals this year is to initiate development around a new version, a version 2.0 of the platform. And Brian and the content committee are working very avidly towards really developing a strategy and an implementation for sharing content across the community. But we still have the future to look forward to. Where are we going from here? And our phase 3, really looking beyond 2014, is to grow to sustainability governed by members, to really stabilize and grow this foundation, this membership, this community, so that it really is vital and sustainable. To implement what is the next version of the platform, to further engage the community, to make a platform that's more robust and more supportable and more extensible as we go forward. We'll be coming together at our annual meeting and revisiting our 3-year plan, which is really to grow to growth and sustainability. We'll continue to focus on geographical centers of excellence. We're hiring a European community manager to augment what Kevin has been doing in North America, and we're beginning to look at that for Asia as well. And then we'll continue to build the programs and resources and infrastructure to ensure the quality and usability of the platform and content around it. So what does the foundation need from you, from the community overall? Become a member, support the foundation, participate in governance and direction, come in and join with us and work with us to build your community. Advocate for the platform. 1.2 is really the launch pad for making tranSMART more ubiquitous throughout our research community. Be an advocate for the platform. Go out and give talks at meetings, speak to your colleagues, proselytize for the platform, advocate for the platform. Participate in our community. The key thing that we're doing is really driving participation. You can see that we had nearly 100 different people participating in our testing individually in our community testing program. So participate in software development, participate in testing, participate in the documentation development that we're doing, the video and scripted development, and provide scientific input. We have the 3C committees, we have these working groups, and you have a management team here that's willing to work with you to enable your participation. And most of all, attend the annual meeting. We have our next annual meeting coming up this time in the States. Next year will be in Europe again. October 14th to 16th. It's in Ann Arbor, Michigan, at the University of Michigan, who's hosting us. We'll have an on-site board meeting for this. We'll have a big celebration for the people that have been the key contributors for version 1.2. It's going to be a great meeting. I encourage you to attend. And if you have an interest in this speaking, we have a few speaking slots open still. We have a few sponsorship slots open still. Contact me or contact Rudy Potenzone, and we'll get you into the schedule as quickly as we can. But please plan on attending the annual meeting. I think it's going to be a fantastic time. It's going to be great to be together with our colleagues again, which we did again, last year in Paris. And I look forward to seeing all of you there. So I'd like to thank everyone who participated today. I'd like to thank Kevin for organizing, Terry for making the machines work on the back end, along with Peter and the whole development team. I'd like to thank Brian for a great MC for the demos, and Rudy for the work that he's done there, and the rest of the team. So with that, I'd like to thank everyone for attending. I'd like to thank you all for your attention, and I look forward to working with everyone in the future and seeing you in Ann Arbor.
Thank you, Keith, and thanks for all the work that you and Kevin have done. And let's just remind everybody that tomorrow at 9 a.m. Eastern Daylight Time, there will be another version of this demonstration activity run out of the IDBS headquarters in London with the Imperial College group with EK presiding. And I'm sure that there'll be some new twists and certainly some new folks. The European folks will be there. And I know many of us here will be logging on, and I encourage others to log on. Part of our challenge is to build bridges across the pond. That's been, one of the things that has actually made the tranSMART Foundation and our activities a big success. And EK and, Peter and Jonas and Case, we wish you all the luck and everyone else tomorrow on that demonstration, and we'll be logged on to show our support. Thanks, everybody.
Fantastic. Thanks, everyone.
Yeah.
Okay, very good.